Hello, everyone. Mr. Fradov again. I am going to go over the 9th chapter, which is on cellular respiration, and this is a difficult chapter. I would honestly say this is the most difficult chapter you're going to have all throughout biology. It deals with how we break down glucose, specifically in our cells. So we're going to go over a couple of different vocab words before we get into the inner workings of cellular respiration. First is the word metabolism. A lot of people think metabolism is just how fast you break down food. It is a part of metabolism, but the metabolism we're talking about just in general is all of the chemical reactions within your body. Specifically when people say like, oh, I have a fast metabolism or, oh, my metabolism's slowing down, they're specifically talking about something called cellular respiration. And this is the process by which glucose is taken in through carbohydrates that you eat and you produce ATP. So your metabolism is a lot more than just cellular respiration. Now, when you eat glucose, you're eating sugar, yes, but you're eating it usually in larger meals like carbohydrates and other macromolecules. You don't technically eat glucose within your proteins because they're not made out of glucose, but you eat a lot of different molecules that you're going to use to break down and to form ATP. Remember, ATP is that energy currency of the cell. Now, a couple of different parts of the mitochondria that we're going to need to know going forward. You have two membranes. You have an inner and outer membrane in this mitochondria. Remember, this is where cellular respiration is going to occur. This is the remember from chapter 7, we talked about how this is where we produce our ATP. Now, the matrix, you can see here, the matrix is the location inside the mitochondria here. This is where a couple of the steps are going to occur. This is like the cytoplasm of the mitochondria. The inner membrane space, this is the area between these two membranes. It's called the inner membrane space. Specifically, this is where we're going to build up hydrogen ions later on. And cristae, these are folds within the inner membrane. This increases surface area of the inner membrane. So you can see here all these little indentions here. We have these folds. This increases surface area. And this is a cross section of the mitochondria. Here's more of a 3D look. Remember, these mitochondria are in our cells, in the cytoplasm of eukaryotic cells, plant and animal. So we're going to need to know the locations here a little bit more in depth going forward. Now, remember ATP, that molecule we used in our last chapter of photosynthesis. ATP is our high energy molecule that we're going to get energy from. It breaks down into ADP in a phosphate group. To reform to rebond these two together, we're going to use energy in the form of food or food energy. So again, ATP has 3 phosphate groups. It breaks down, and that's where we get our energy from. Then to form ADP and a phosphate group back together, we need energy from food. Now, if you see here, our inputs are out and our outputs are, they're very similar to photosynthesis. Our inputs, glucose and oxygen. If you remember, these were our outputs of photosynthesis. Our outputs of cellular respiration, carbon dioxide, water, and energy. These were our inputs for photosynthesis. So these two reactions are opposite reactions, or they're reversed reactions, kind of. What goes into one comes out the other, and vice versa. So what goes into plants and is what we breathe out and aspire. What plants give off, glucose and oxygen, is what we take in. You can see here carbon dioxide and water going into photosynthesis. It's also using sunlight to produce glucose and oxygen. Glucose and oxygen go into cellular respiration, and we get that energy out of those two molecules in the form of ATP. So essentially, the ATP energy that we're getting comes from the sun. Not directly, but that transfer energy starts at the sun. All right, this is the chemical equation of cellular respiration, and if you notice, this is just the opposite reaction of photosynthesis. So again, in photosynthesis, we took in carbon dioxide, water, and energy, and we produced glucose and oxygen. In cellular respiration, we're producing glucose and oxygen, we're taking in glucose and oxygen, and we're producing carbon dioxide, water, and energy. Except in this form, ATP is our energy source. You can again see when we breathe out oxygen, we breathe out CO2, my bad. We breathe out CO2, plants take that in, they breathe out oxygen, and we take that in. Now, the 4 steps of cellular respiration are glycolysis, the prep step, Krebs cycle, and electron transport chain. This is where it does get a little bit more difficult. We will talk a little bit more in depth about each of these. I don't have you write these down specifically at the beginning because we look at each one of them a little bit more in depth. And let's take a look right now. So remember that in glycolysis, that glyco means sugar and lysis means to break. So this is the initial breakdown of sugar. This occurs in the cytoplasm. 2 ATP are used up first to break down this molecule. So we have this molecule, glucose. I only show 6 carbons here because the glucose molecule is pretty complex. I only care about in this class where the carbons are. So in this case, again, you got to use money to, you got to make, spend money to make money. That's the analogy I use. You got to use 2 ATP to break these down. So off the bat, we're already negative 2 ATP, and that's not good. It's like when you start a business, you have to spend your own money to get that business started. So to get this going, we have to use 2 ATP. So that one glucose molecule that has 6 carbon molecules is going to be broken down eventually into 2 3-carbon molecules. And we produce 4 ATP and 2 NADHs. Those 4 ATP are right here, and those 2 NADHs are right here as well. Now, the confusing part about this is you do produce 4 ATP, but you only actually produce 2 net total ATP. The reason being is, remember, we already used 2 ATP. So like, this is an example. If you are starting a lemonade stand and you spend $100 creating that stand and you make $200 on the first day, you really only profited $100 because you already spent $100. You have to kind of spend money in this to make money. It's kind of like a business. You have to spend money to make money. So at the end of glycolysis, we made 2 ATP, we made 2 NADHs, and we have 2 pyruvate molecules, each that have 3 carbons. All right. The prep step. This occurs in the matrix of the mitochondria. And what happens is the pyruvate molecules are both going to move to the matrix inside the mitochondria. So again, this is the cytosol or the cytoplasm. It's going to move these pyruvate molecules into the mitochondria. These pyruvate molecules are converted into something called acetyl-CoA. You can see this molecule, acetyl-CoA. It looks really complicated. Just understand it has 2 carbons. This produces CO2. It produces another NADH. And we're going to come back to NADH later on. And a coenzyme A is actually added here. So you can see coenzyme A. This occurs twice. Why does this occur twice? One pyruvate molecule is going to go through this process, and then the next pyruvate molecule is going to go through this process. So you have to essentially double everything. We double the amount of CO2, double the amount of NADH, and we double the amount of coenzyme A that we are using. So now we're through 3 steps. We're going to get through the 2 steps. Now we're going to go through that 3rd step. Now the 3rd step is arguably one of the most difficult steps, but again, I don't have you learning too much about it. This occurs again in the matrix of the mitochondria. Acetyl-CoA is going to enter this cycle, and you can see how this cycle is a kind of like a, I want to say a merry-go-round, but like a cycle that goes around and around. We produce 3 NADHs, we produce an FADH2, and we produce 2 CO2s and an ATP. So there's a lot of reactions going on in this cycle. I mean, there's a lot of reactions. You don't need to know about any of those reactions, okay? What we do need to know is 3 NADH are produced, 1 FADH2, 2 CO2s, and 2 ATPs. I know there's a lot of words here, and I know it sounds very confusing. Try to memorize just where each of these are being produced at. Now remember, per acetyl-CoA, we're making each one of these. So we have to double it because we have 2 acetyl-CoAs coming in from the prep step, and that's because we have 2 pyruvates going into the prep step. So this occurs twice. So essentially we make I'm sorry. This should be 3. Make sure I have a little mistake here. I changed this today. This should be 3 here. So this should be 3 NADH, 1 FADH2, 2 CO2s, and 1 ATP. We have to double everything here in the Krebs cycle. This is what it actually looks like. I'm just showing you this just to give you a little bit of an understanding of how complicated this can get. I don't really care if you even, kind of look at this. This is just to show you, hey, this is pretty complex and there's a lot more going on here. All you need to know is acetyl-CoA is coming in here. It's producing 3 NADHs, 1 FADH2, an ATP molecule, and where are my CO2s? Oh, right here, CO2 molecules. Now, we've been talking a lot about the NADHs and the FADH2s. What you need to know is these FADHs and NADH2s are like electron and hydrogen ion carriers. They take hydrogen ions and electrons to different places called the electron transport chain. And we're going to get to that here in a little bit. Our last step here of the cellular respiration is called the electron transport chain, or if you see it, it's called the ETC for short. This occurs along the inner membrane of the mitochondria, and we just talked about those NADH and FADH2 molecules. What's going to happen is they're going to deliver electrons and hydrogen ions to these membrane proteins. What these membrane proteins do is they dump off the electrons, they dump off hydrogen ions, and they force the hydrogen ions into that inner membrane space. What that does is it creates this high concentration of hydrogen ions here and a low concentration of hydrogen ions here. So in the inner membrane space, we have a lot of hydrogen ions. In the matrix, we don't have a lot of hydrogen ions. That creates a concentration gradient of high and low concentrations between the intermembrane space and the matrix. What happens at this step specifically is ATP synthase is going to make ATP. And I'm going to explain that next. Before we get there, though, is this here, this process needs oxygen. That's why they call this an aerobic process. What happens is this oxygen is going to be bonded with hydrogen ions, electrons, and the oxygen to form water. Without oxygen, nothing else happens here. So this is part of the reason, not only reason, but this is part of the reason why when you don't have oxygen, you're going to die, because you don't have that oxygen. This is that final electron acceptor. Remember the spaces that we talked about in the mitochondria. We have our inner membrane, we have our outer membrane, we have our intermembrane space, and we have our matrix here. This is all occurring here along the inner membrane of the mitochondria. Now, what's actually happening is the ATP synthase is having hydrogen ions move through it, and that movement of hydrogen ions through the ATP synthase is going to bond a phosphate group to a DP, making it a TP. This process is pretty cool, and I'm pretty sure I have an image to show you, but what actually happens is these hydrogen ions move through, turning this ATP synthase, bonding a phosphate group to a DP, making it ATP. There's a total of 34 ATP produced at this step per glucose molecule. And if you remember, we first talked about how there are 38 ATP produced per glucose molecule. 34 of them come from this step, and 4 of them come from the first couple steps. So the majority of phosphorylation of ATP, or the production of ATP, occurs at this step. I told you there's a lot of big words here. You need to know overarching concepts. So don't get too nervous about this chapter. It's not that bad once you look at it. And here's that ATP synthase turning, producing ATP. And I know it looks kind of weird and just crazy but this occurs in all of your mitochondria of your cells. So again, we have this hydrogen ion concentration. It's going to move through the ATP synthase. It's going to turn that ATP synthase. That ATP synthase, as it's turning, is going to bond a phosphate group to an ADP molecule, making it ATP, and a majority of our ATP is produced here. Now, this is an aerobic process which requires oxygen. As I told you there before, it requires oxygen, but it produces a lot of ATP. One place where we don't need oxygen is called an anaerobic process, and it does still produce ATP, but very small amounts. I always give the analogy of if you're running like a really long race and you don't have oxygen, you feel really tired because your body's not producing enough ATP. That's because it's going through an anaerobic process, which means, again, you don't have enough ATP, or you don't have enough oxygen in your body, so you're producing less amounts or smaller amounts of ATP. Let me show you what that kind of looks like. All right, so again, an anaerobic respiration doesn't use oxygen. This is not an efficient method of using and producing ATP. It's also known as fermentation. There's two different types. We have lactic acid fermentation, which goes on in your body, and we have alcohol fermentation, which goes on in bacteria. In lactic acid fermentation, what happens is your body stops going through the prep step, the Krebs cycle, and the electron transport chain, and the only step that occurs is glycolysis. That means only 2 ATP will be produced. So normally with oxygen, your body goes through the rest of cellular respiration. Without oxygen, your body goes through fermentation and only produces 2 ATP molecules. That's because the only step here you can see glycolysis occurring. I got to sneeze, I'm sorry. Woo, sorry. So anyways, back to fermentation. So glycolysis still occurs, but what happens is these pyruvate molecules are going to get changed into something called lactate or lactic acid. And that's just to free up more NAD so this process can still occur. Again, you're only producing 2 ATP, which is not a lot of ATP, but you're still at least making a little bit of ATP. The other type of fermentation is called alcoholic fermentation, and this is how we make a lot of our wines and beers. What happens is you have to have one of these cauldrons, and you have to take out all of the oxygen. This creates a alcohol fermentation in the yeast or the bacteria that are being used, and this is what also makes bread, cheeses, wines, beers, and the byproduct there is what is actually making the alcoholic beverages alcoholic. It's an alcohol molecule. You can see this process. We still have glycolysis occurring, but the end product is an ethanol molecule, not lactic acid. So again, the last part here, I just want you to understand the difference between aerobic and anaerobic respiration. In aerobic respiration, we go through the glycolysis, the prep step, Krebs cycle, and the electron transport chain. In the anaerobic respiration, we're only going to go through that glycolysis, which we only get 2 ATP. So we get a lot more ATP in the aerobic respiration, not a lot in the anaerobic respiration. Our products here are carbon dioxide and water. Our products here in anaerobic respiration are lactic acid and alcohol, or one of those two, not both. One of those two.